I'm Montana Smith. I am a scientist at the Environmental Molecular Sciences Lab at Pacific Northwest National Lab. I work primarily on sample metadata collection and curation for EMSL user projects, as well as on the NMDC. And today I am joined by Shankar Subramaniam. I hope I said that right. Lynn Schrimmel, Scott Jackson, and Max Saito. Shankar is from UC San Diego. Lynn is a member of the Genomic Standards Consortium. Scott from the National Institute for Standards and Technology, and Mock with the Woods Hole Oceanographic Institution. And so today we'll be talking about sample metadata or metadata standards in general and the importance of them and how to develop them. And so I want to start with just kind of a few overviews of the NMDC and FAIR and metadata. So the NMDC is the National Microbiome Data Collaborative, and the mission for the NMDC is to provide a gateway to FAIR multi-omics microbiome data, leveraging best practices to data curation and processing. So it seeks to provide a place where researchers can share their data, find multi-omics data from various microbiome studies, and understand the data and samples that they're looking at. The NMDC also provides standardized workflows for data analysis. So when a researcher finds data on the NMDC, they can work with the results in confidence in knowing that they've been processed or analyzed consistently, or that they at least understand how that's happened. We're here today for the Community Conversation Series, which is a series of discussions about various topics that are important to samples, data management, data curation, and science in general. So today's community conversation, as I said, will be about curation and standardization of metadata for different samples and omics processes. So metadata comes in all shapes and sizes. Within the NMDC, when we talk about metadata, we've placed these into categories to help people understand what they're talking about at what stage. And so here you can see that we have, okay, so metadata comes in all shapes and sizes within the NMDC when we talk about it. We have sample metadata, which describes the environmental material or the biosample that you've collected. There's preparation metadata, which describes what you did to the sample to generate biomolecules or an analytical sample. And that will give rise to what goes onto the instrument, which then gives rise to your data. There's also process metadata and feature metadata, which describe how the data is analyzed or used. So when we use why do we care about metadata for each of these categories, and why do we care that it's standardized? Well, if you've ever tried to work with data that's been generated from someone else, or if it's data that you're trying to match within even your own experiment across analytical types, it can quickly become a jumbled mess, and science becomes unnecessarily even harder. When we use standardized metadata, the context and connections across samples, data types, and experiments become more clear. Identifying standardized sample metadata about the when, where, and what of the biosample ensures that you and anyone who might use that data later on understands what the samples are and what could be causing biological differences. Standardized preparation metadata and data processes are needed to know how a biosample is prepped or extracted and how to compare across the data types. Without it, large-scale analyses or modeling would not be possible. And using standardized method at each of these steps ensures details are not missed. And when a factor or detail is called one thing from source A, you know how it's similar or different to something from source B. Accomplishing standardized metadata in each of these steps also allows you to adhere to FAIR data principles, and it makes your data discoverable, accessible, and comparable, as well as persistent with assigned data DOIs, which will also increase the scientific impact of your work and how recognized it can be. So again, before I hand this off to our first panelist, Lynn, I want to remind you all to please add your questions and discussion points to the chat or the shared notes doc. And so with that, hopefully Lynn will have less technology difficulties than I will. Thanks.
Hello, I just need to have the slides pulled up. Great. Okay. Thank you all for coming and listening about metadata today. So this is always something that's very near and dear to my heart. I lead the Genomic Standards Consortium. We've been running since about 2005. So it's quite a long-term effort by, I'll show you a large number of community members over time, to really think about, well, how you capture the data, how do you communicate it, how do you implement it into your systems, and then how, in the end, how do we really share all this information to do better science and have ongoing analyses. So today I was asked to talk about the history of standards and how do you develop standards for samples. And this is actually a really good question because I get asked this quite often from new research groups that want to build a standard. Could we go to the next slide? Great. So I'm going to talk about the history just a little bit. On the slide, I'm showing you kind of the walkthrough over time of genomics. But let's go back just a little bit earlier to genes. So back in the 80s into 90s, into early 2000s, we all thought about genes a lot. Gene nomenclature committees. That's where standards really were starting to be initiated. And as we've walked through this timeline of genomics, our standards have evolved to meet the new technology needs, to look at new environments and new challenges. Next slide. Now, just talking a little bit about developing standards for samples. This is a generic slide intended to really give you a peek into what communities go through when they're developing a standard. I have to say, honestly, when we started the consortium, we're getting almost 20 years now, we didn't really think about this process, but this is very much what we've gone through. And now we actually help other communities do this. So I think the big takeaway from this slide is to think about when you're facing a common problem, how do you bring a community together? How do you bring others into your community so you can grow and evolve and bring their voices in and their perspectives? And then as you go to the right part of the slide, you're seeing what the process is involved, from bringing community members together, identifying goals to work on together, figuring out how you're going to work together, what will the physical work look like, how to have all the opinions brought together, and then towards the end of the effort, how do you make this resource, this standard you developed, to be utilized by others and to be utilized within your own project. So this is really much a kind of a step-by-step plan, and we work with numerous groups to do this so that they can bring together and establish this list of terms that they want to collect are important for their science. Okay, next slide. Again, I want to focus on the point of a common goal. When we all are working on our own research projects, we come together, maybe with colleagues who are of a similar topic, and we write a paper together, or write a grant together. Developing standards tends to be a little bit larger of a project where you're reaching out to people that you never knew before, you wouldn't probably have had contact with necessarily, but they have a stake in the game. They're interested in a similar topic. We would come together on these common challenges, and the reason I have the leafcutter ants here, they work together as a community to build something much larger than themselves, and that's very much what this kind of collaborative effort does. When you're trying to build standards and to change a community's way of communicating about the data, and then to share that data far beyond the current research studies. Okay, we can go to the next slide. So I'm going to introduce here the Genomic Standards Consortium. At the top of the slide, you'll see our mission, and our website is there. Really, it did get cut off just a bit, I think, some of the text, but that's fine, from when we converted to a group slide deck. So what I wanted to tell you about the mission is that we've always been focused on this common goal of making data discoverable within genomics across the different types of genomics that have evolved over time and across the different types of environments. It is very much a communication, this type of consortium, working together, bringing in new stakeholders, doing outreach with journals, with other research communities, so that we can drive the science forward, support ongoing efforts. And as you can see and then the news item on the slide, we just had our annual meeting in French Polynesia. That we do these annual meetings to bring in new groups each year to identify and really suss out the standards needs for different communities, and we continue to do this on an ongoing annual basis. We also have monthly meetings for some of our working groups, so if you want to build standards, please contact me and we can put you together with that. Okay, next slide. this slide is really showing you the guts of what our genomic standards have accomplished and produced in the last almost 20 years. So we started out looking just at genomes in water samples from the English Channel and soil samples, and it's evolved over time. As you can see, the different genomic checklists, we call them at the top. These are different types of genomic data. One example is the minimum information about a metagenomic assembled genome. So a metagenome assembled genome is something much newer in our world of genome science. But as this work has evolved, we continue to develop these standards. And also at the bottom of the slide, you're seeing the various environmental packages. Now, this is an extension of the initial work that allows us to capture data pertinent to particular types of environments where people are doing studies, like most recently with all the COVID-19 outbreaks and universities looking at the sewage samples across their campuses. Again, some of these packages, we have a sewage package in here. And water package, it would be helpful as well to again look at how we capture this data. And then the last update for this slide is that we did just send out, it was even just a week ago, our latest annual formal release. This goes out to all of the data repositories that host all this data. And in this particular update, we have a number of packages for food, working with the FDA. So from production all the way in the field, farms, through the production facilities and to the grocery stores, and then agricultural microbiome community and symbiotic community. And we continue to add new ones each year based on community needs. And then I think if you could click one more. Great. I'll just give you a bit of a timeline of what standards development as a community looks like. And I'm not going through all the details of the slide, but if you look from left to right, you can see in these 17 plus years we've been working, we did that same formula I mentioned earlier. We developed a community, grew it, evolved it, continue to expand, and also to reassess what was our place in this community. How can we best serve what the community needed to, again, build on the data that's being developed and the science is evolving. Okay, we can go on. And I just want to include this slide. These are the current board members as of about a week ago. We just elected 3 new ones, but as about a week ago, showing the diversity of networks you build with building this kind of standards effort. As I mentioned before, you really need to think about the broad spectrum of people that will be involved and type of data you're doing. But because of the age of our consortium and how we've developed, you also have tool development and resources. So you can see a number of them listed on this site, where members of our community are also involved in those aspects of genome science, which are critical aspects to have a standard utilized across many needs. Okay, and then I think we're near our last slide. Yeah, I just want to show a little bit about the impact potentially of a standard. So these are just a snapshot of a number of different journal articles that we've published over time. Just again, it's another way of sharing your information when you're developing a standard to a broader community beyond your own. Okay, I think that's it. Okay, there we go. Thank you very much.
Great. Thank you so much, Lynn. So now we'll move on to Scott. And again, if you have any questions for Lynn, please stick them into the chat or into the shared notes doc.
Okay. Thanks, everyone. Can you hear me and see me or see the slides? Yep.
Yep.
Okay, thanks for the invitation. So I'm Scott Jackson. I lead the microbiology group at NIST, the National Institute of Standards and Technology, CLIC. I feel obligated to introduce NIST because I know a lot of times people are not familiar with NIST. We're a non-regulatory federal agency in the Department of Commerce. We're the oldest technical institute in the federal government dating back to 1901. We have 2 campuses, one in Gaithersburg, Maryland, and the other in Boulder, Colorado. The pictures are on the right. I bet you can guess which one is in Gaithersburg and which one's in Boulder. About 5,000 scientists and engineers strong. And to our placement in the Department of Commerce, our mission is to promote U.S. innovation and industrial competitiveness by advancing measurement science standards and technology. And in the biosciences division at NIST, we talk a lot about the bioeconomy and how we keep the United States competitive in the global market. Click, next slide. Okay, so next-gen sequencing-based metagenomics. It's really the foundation for microbiome science. I like to think of it as the 21st century microscope because just as the microscope did when it was discovered in the 1600s, it allowed us to see things that we couldn't see before. And that's exactly what the next-gen sequencing instrument does today. It allows us to see things that we couldn't see before. So this is the measurement workflow of a metagenomic analysis workflow. This is what you would do if you're in the laboratory. So, step one, sample collection and storage. Move on to your nucleic acid extraction, prepare your library, sequence DNA, do some preliminary data filtering, and then the statistical analysis and reporting. And click, please. My point here is that there's bias in every step of this measurement process, from how you collect and store your data to how you analyze and interpret your results, and everything in between. And there's a lot in between. I could spend 45 minutes on this one slide just talking about the methodological variability and how it impacts the results. So how in the world do we harmonize on these microbiome measurements in a way that two labs across the country or across the world can measure the same sample and get the same result? We're far from that right now. Click. And that's really one of the goal of our work at NIST is to harmonize these measurements, develop standards so we have better reproducibility and less interlab variability, I guess. This slide just shows, one of the ways we operate. Being in the Department of Commerce, we recognize industry as being an important stakeholder. Those biotech, biopharma companies that are developing the next new modality of drugs that are targeting your microbiome, companies developing microbiome diagnostic type testing, measurement assurance strategies for these types of industries are very important. But at the same time, these industries are regulated by FDA. So we work closely with FDA to develop industry standards that are then used by industry to develop products that are regulated by the FDA. So you can see sort of the cycle there that, we work with industry and we work with FDA, and FDA works with industry and FDA works with us. And it's all around building industry standards so that the FDA can make more informed regulatory decisions on the products that they regulate. Click. Something didn't come through. So just this morning, we had a internal group meeting at NIST, and we went over all the projects that are going on, and we have about 15 ongoing projects. Unfortunately, today I only have time to tell you about one. So one of the NIST standards, reference materials that we started developing about 5 years ago and just finished last year, was what's called our mixed pathogen DNA reference material. And if this cartoon had come through as it was supposed to, you would see that these different colored pieces of DNA represent different pathogenic genomic signatures from different pathogens. And you can make mixtures of these different genomic DNAs, both quantitatively and qualitatively. That is, we know which genomes we can mix together in the tube, and we know their relative abundance. So essentially, we can make ground truth, complex microbiome samples that then allow us to benchmark our measurements. Click. So once you've produced your complex mixture of genomic DNA that is ground truth, you can run it through your favorite next-gen sequencing platform. Click. And the results that come out, you can compare the expected result to the measured result. So this is, click, please. This is known as RM8376. As I said, it came out about last year. The picture to the right is one of our lead scientists, Jason Kroll, standing in front of the refrigerator full of this material. You can call up, or not call up, but you can go to the NIST website and purchase this material for use in your own lab. And there's a tiny URL at the bottom there that I mentioned. So, this is one example. As I mentioned earlier, we've got about 15 ongoing projects now. This was one of them. And all of our ongoing projects are focused on microbiome measurement assurance strategies. And I'll just give you a hint at another project that's ongoing. We're developing a human fecal reference material, so standardized human fecal material that can be used to perform interlab studies and benchmark new measurements, develop new measurements. And just like we produced RM8376 here, the mixed pathogen DNA, pretty soon we'll be coming out with a human fecal, aka gut microbiome material. Click. This RM8376 that I mentioned, it's mixed pathogen DNA. Yes, we make the material, we make it publicly available, and that's fine and dandy, but more interesting on our side is using the material and assessing its capabilities. So this slide, I just want to show you an example. Using our own RM8376, we made a lot of complex mixtures of these genomic DNAs. And looked at, the expected result versus the measured result to see how well we do. These are metagenomic samples. So when we run them, when we analyze the data, we want to run them through a taxonomic profiling tool. And one of the things I want to show you here is that 3 different taxonomic profiling tools that we use, that is Centrifuge, Gotcha, and Metaflan, gave us strikingly different results. So consider this is the same raw data set that came off the, same sequencing instrument, but we just ran it through 3 different analysis tools, got 3 dramatically different results, just to show you the methodological variability and how it impacts your result. Click. Moving on with that, I just mentioned 3 taxonomic profiling tools. There are well over 100 that exist. So 100 bioinformatic tools that have been developed seemingly for the same purpose, that is to taxonomic profile your metagenomic sample. They all use different algorithms, different statistics, different assumptions, and different reference genome databases, and they will all give you different results. So here's an example of a methodological variability that will impact your results. And I just have, I think, a couple more slides quickly. Click. Profiling the profile. So which of these taxonomic profiling tools work the best? There have been a number of benchmarking studies that have been performed. One that I like to point to is this one by Lindgren and Gartner. Click. Where they looked at this list of different taxonomic profiling tools. You can see on the left, Clark, EBI, Gotcha, et cetera. And they looked at different performance metrics like number of true positives, false positives, true negatives, false negatives, sensitivity, specificity, positive predicted value, negative predicted value, on and on. And click, please. The last thing I just want to point out to this, if any of these tools worked perfectly, then all 5 of these bars would be pushed to the right. And as you can see, depending on the performance metric, some work better than others. None of them are perfect. So when you're deciding which taxonomic profiling tool should I use, you need to look at data like this and assess what is my goal? What type of samples am I looking at? Is sensitivity more important than specificity, or is specificity more important than sensitivity? These are the types of questions that you should ask yourself, and look at these type of benchmarking studies and assess which tool is right for you. Click. One more study that I'll mention was done by Chris Mason's lab at Weill Cornell School of Medicine. This one was actually led up by Alexa McIntyre, shown in the bottom there. This is a figure that I grabbed from their paper, and I grabbed it because it's quite alarming what they're showing here. So again, using one data set, they ran it through these 20 different profiling tools, and all they're showing here is the number of species that were detected using that tool. And notice on the x-axis, this is a log scale. Some tools detected tens of species, some tools detected thousands of species, and these are all being run on the same raw data set. So it shows you the impact of methodological variability in your choice of taxonomic profiling tools. And that's just one step in a very long step, in a very long process of measurements. Okay, click. I'll mention quickly IMSA. IMSA is the International Microbiome and Multi-Omics Standards Alliance. We started it back in 2015, 2016. Click. It's an open consortium. You can all welcome to join. Just visit imsa.org. I do want to ask you all to save the date if you would like to join us in our first IMSA workshop being hosted later this year, August 23rd through 25th. It will be a hybrid event, both in person and virtually. It'll be on our NIST Colorado campus. Again, the website is imsa.org. You can find out all you want about IMSA. Click. And we want you to join IMSA. Go to imsa.org. It's free. It's a community resource to discuss measurement assurance strategies and standards for microbiomes. Click. That's me.
Great. Thank you so much, Scott. And now we'll move on to Mac.
Great. Well, it's great to be here. I'm at the Woods Hole Oceanographic Institution, and I'm primarily a data producer of proteomic data. And ocean metaproteomic data in particular. And just as a brief introduction, the measurement of proteins in a diverse biological community is how we define metaproteomics. And it's really valuable and useful and complementary to other omic techniques because it gives us information about ecosystem function and health. But it's actually really different from a lot of nucleic acid type measurements, genomics types measurements, because the basal unit is mass spectra. And that's shown here on the right, where we have our individual mass spectra, tandem mass specs, our precursor masses, and below that are fragment masses. But you know, when this is compiled, we're able to get these really useful data sets that show nitrogen stress and iron stress, for example, of different microbes in the oceans. Next slide, please. And so a few years ago, we gathered a community of ocean proteomic researchers, as well as a lot of microbiome colleagues as well, who we knew were doing metaproteomics. And we had a workshop to talk about best practices for data sharing. This was a really exciting workshop. A lot of us hadn't met before, but we were able to focus on a lot of the challenges in our field as we reviewed our field, and that includes sample collection, analysis, results, and interpretation and dissemination. And a lot of this, of course, there's a tremendous amount of metadata and informatics challenges that are associated with metaproteomics, just like other metaomics fields. And we compiled these results in this publication that's available both in open access at the Journal of Proteome Research, as well as on the Ocean Best Practices website as well. Next slide, please. And part of the motivation for organizing the community is that we're trying to enable global data sharing efforts. We know that individual researchers, we can't all cover the entire planet and the entire ocean together individually, but together we can get enough coverage to actually make sense of what's happening in the environment. And so we've been developing portals such as the Ocean Protein Portal to do this, and which can take data from a variety of, or anyone who really wants to submit, and allows non-data generators, non-proteomic experts to go in and start examining the data, as well as answering their own hypotheses and exploring how the oceans work, as well as using it for education purposes as well. We've had a number of classrooms start to try to use it in their classroom settings. Next slide, please. And another part of the motivation is that the international community has been working together for a few years now to try and launch a new global ocean microbial biogeochemistry program called Biogeoscapes. And it's essentially studying ocean metabolism and nutrient cycles on a changing planet, with the aim of trying to do true multi-omic studies, as well as combining with a lot of chemical and biological discrete measurements. And so it's a really exciting effort. We just found out this week that a big management proposal for international networking was funded. So we're going to be doing a data management workshop in the coming years. And so if any of this is interesting to you, I encourage you to go to that website, biogeoscapes.org, and sign up for the mailing list so you can keep in touch. And we would certainly love for people in this community to engage with us, because it's going to be a tremendous informatic effort. Next slide, please. And so in this workshop that we had on ocean metaproteomics metadata, we got into the nitty-gritty of defining what our metadata needs were, and came up with a table of best practices for various parameters, very much like the slide Montana showed earlier. Talking about the project metadata that we need, as well as contextual metadata for the samples. And of course, this is really important and what makes environmental work distinct from a lot of medical work. We need to know the latitude, longitude, sample depth, sampling type of approaches, as well as availability for environmental data. There's also information about the processing of the data and mass spectrometry methods, as well as data analysis methods that are needed. Next slide, please. To dig into a little bit more on the acquisition and data analysis metadata, I think this is actually something also that is particularly important for proteomics, in that there's the way the machine is set up really makes a huge difference in the kind of data you collect. And so documenting that methodological metadata is crucially important to interpreting the data. And so if we're trying to assemble data from a variety of users, being able to interpret them correctly really requires documenting this well. Next slide, please. And then another aspect that's really important that we realized was the data types that are needed for data sharing. So it's not just the raw mass spectra files as well as process files such as protein and peptide identifications and their quantitation numbers, but also the sequence data that we use to generate this data. In this case, the FASTAs of the amino acids. Most often, we use metagenomic data or metatranscriptomic data to do our peptide to spectrum mapping, where we convert those spectral units that I mentioned into the sequences. They rely on curated, usually very specific and maybe sample specific metagenomic databases. And so making sure that information is captured and that data is collected and where to find it is really important for reproducibility and interpretation of the data. Next slide, please. And just to wrap up too, we've just been completing an ocean metaproteome intercomparison effort. This past fall, we had a workshop with these colleagues shown here. And one of the things that we've realized in trying to put this together is that we need new standards on how to, on what kind of data output formats we're using. It was actually very difficult to collate and compare data sets from different pipelines because the formats were just so different, and in many cases, the approaches were different too. And then just to conclude, we could, looking forward, there's a lot of resources that we can leverage. We realize we're not alone in this. There's a wonderful proteome exchange network of repositories out there for proteomic and mass spectrometry data. I'm also a member of the Biological Chemical Oceanography Data Management Office, or BICO-DEMO, which hosts a lot of important discrete measurements in the oceans, and we're encouraging people to put in links out to sequence repositories to connect that to environmental data sets. There's also this growing biogeoscapes community, as well as the EarthCube and NMDC and Human Proteome and other community resources that are out there. Next slide. So thank you very much for your time.
Thank you so much. And now we'll move on to Shankar.
Hello, everyone. It's really a pleasure to talk with all of you. My name is Shankar Subramaniam. I'm at the University of California, San Diego, and my research interests are in systems biology and systems medicine. We do all kinds of omics and data. I live with data. I also run the National Metabolomics Data Repository, which is something I'm going to tell you a little bit about today. Next slide, please. So my goal here to tell you about, in fact, the challenging question that was posed was the role of metadata and the importance of metadata and how it helps in terms of making FAIR as a FAIR entity. And I'm going to tell you a little bit about it. Most of you know this FAIR is findable, accessible, interoperable, and reusable deposition of data. And I'm going to tell you, let's go to the next slide. I went to the GoFair site, which says that fairness is defined by GoFair with these criteria. And if you go to the next slide, it tells you how many times, no, I'm sorry, go back to the one. Please go back one. I thought I had a slide which had highlighted the colors, but maybe it didn't come through. Okay. So if you look at I just asked a simple question. If you ask the question, how many times metadata occurs across these categories that GoFair defines as being required, and I did a wordgram of the metadata, occurrence of metadata in each of these categories of findable, accessible, interoperable, and reusable. And the next slide gives you the number of times metadata occurs. Next slide, please. You can see that the metadata is the most dominant word in this entire set of documents. And I'm going to explain to you why it is so important. And without metadata, data can become irrelevant. And this is what I want to point out to you in the next few slides. Next slide, please. So I run the National Metabolomics Data Repository, and we run this thing called the Metabolomics Workbench. And the Metabolomics Workbench, which now has over 2,000 studies. This was an old slide that I took in 2020. Today, we have more than 2,000 studies encompassing over several millions of metabolites. And this is the Workbench site. I gave you a URL for this. And I'm going to show you how we developed this Workbench to be completely fair. If you go to the next slide. Metabolomics Workbench is fair. We assign DOIs to each submitted data set and study so that it can be tracked and monitored and followed and linked. And we use schema.org formatted metadata. We embedded that in the Metabolomics Workbench so that it can be not only our data and our metadata, but the whole scheme of having data and metadata can be accessed through schema.org. And I'll tell you in a few minutes about that. And you can, in fact, what is the benefit of having schema.org is that it allows all search engines to discover your data structure. Then we also link to the Metabolome Exchange, which I was one of the creators of this thing called the Metabolome Exchange, which is maintained by the European Bioinformatics Institute, including the Omics Discovery Index, which provides links to all the metabolomics data across European studies, including UK and Netherlands, and the US studies, which is the Metabolomics Workbench. And so this findability is very high with the metabolomics data because we access it through all these different mechanisms. Accessibility. To make it really accessible, we provide on the website a completely user-friendly, seamless access options. And I'll show you in one slide as I go along how this is so trivial and easy for bench biologists or bench biomedical researchers to access in a very easy way. In addition, we provide a REST API for people who want to download our data, for accessing the data, to access it in multiple formats. And again, I'll tell you a little bit about the REST API in just a second. Our entire data is interoperable. We use community standards of MWTab to describe the data. And so you can download all the data using the MWTab format. We document it in fairsharing.org. So fairsharing actually is adopted our MWTab format as a universal format for metabolomics type data. We have multiple positive outcomes we developed a new standardized nomenclature called RefMet. RefMet allows us to harmonize metabolomics data, including microbiome metabolomics data, across all existing resources. And I'll tell you a little bit more about this as I go along. Finally, we are reusable. You can download and process data from NMDR. We provide you a list of hundreds of tools on the workbench so you can interactively analyze your own data as well as the data present in the Metabolomics Workbench in a completely seamless and interesting manner. I'll give you a little bit more about it. And most importantly, we provide you Jupyter notebooks, which illustrate how to use and reuse the data. So this is how the Metabolomics Workbench is very fair. In the next slides, very quickly, I will go through how we make all of this happen. And please go to the next slide. I'll be very fast in telling you. Schema.org is a collaborative community activity with a mission to create, maintain, and promote schemas. And for example, embedding schema.org metadata allows you to be indexed by Google dataset search, and you can immediately identify, any type of, study that you want to look at in a very rapid manner. So I would urge all those data creators, resource generators to really adopt schema.org to make life seamless for the community to use. Next slide, please. I'm going to show you about REST. mentioned to you about REST service API document. We provide a 26-page PDF file of API information. So you can really use this. If you're a programmer, you can play with it, you can use it, you can manipulate it in a way that is interoperable with your own data that exists. And if you're an end user, you can just download it using the REST service web page, and you can download any data sets or tools that we currently provide on the workbench. Next slide, please. Example of customized data retrieval using REST API. Here's an example. You can get REST information from untargeted metabolomic studies. You can extract the data for conditions of interest in a JSON format. And then the JSON is often used as an input for meta-XCMS analysis. So we provide you with a very convenient format for you to use it. Again, this illustrates to you the importance of interoperability of the data. Next slide, please. In addition, for biologists, somebody like me who really is interested in actual biology, I want to view my data in formats that makes it life really easy. For example, I can view my data in terms of the importance of metabolites in a given disease or in a given species or in a given condition. And I can look at class enrichment in an almost trivial manner. I can build volcano plots. I can build bar plots. All of this is accessible through a one-point click through the Metabolomics Workbench, as I'll show you in one slide as I go along. Next slide, please. So this is an example of what I told you. I can use, for example, parse my data for any given data set into species-specific measurements, sample source-specific measurements, disease-specific measurements, pathway-specific measurements. So it's really versatile in terms of allowing you to look at a glimpse or a single glance, parsing of all the metabolomics data available on the workbench into all these different categories at a glance. And you can drill down. This is all web accessible and linkable. You can drill down into a given species. You can look at all the studies. You can take a given study and look at how it relates to pathways, all those kinds of very detailed information. Next slide, please. We developed this thing called RefMet. Before we came into the picture in the early 2000, the last reference standards that existed for metabolites was the late 1960s and early 1970s. In fact, at that time, think about it now, when you call a substance like sugar, sugar can be glucose, it can be fructose, it can be anything. It can be, you can call it as a D-glucose, you can call it as a, multiple nomenclatures you can provide, but how will somebody not be confused? So we created something called RefMet, which is a reference list of metabolite names. And this is really universal now. We have 200,000 metabolite species in RefMet. It's become an adopted standard for the community at large. This is another thing that makes data interoperable and metadata linkable to data. Next slide, please. So here's how we do classification. We take a metabolite study, we classify to lipids and non-lipids, and then we classify to RefMet. We make this completely accessible to the community. So go ahead and download this from our workbench if you want to categorize your own studies. Next slide, please. So this is a parsing of all the studies. Right now we have over 2,000 studies. This is an old slide. We have 220,000 metabolite species across the studies, and we parse them into what type of metabolites are they? Organic acids, glycerolipids, fatty acids, carbohydrates, stereolipids, etc. And we provide this dynamic classification, and you can click on each of these and dive into more into details in each study. Next slide, please. So we also do the classification to look at class-specific data analysis. You can say, for example, I'm only interested in glycolipids or sphingolipids. And you can go in and map all the sphingolipids, and you can map them to pathways. You can map them to various significance plots. You can say, these are the 4 sphingolipids that are most important for my diabetes study and things like that. So this really helps you navigate your data in an interoperable manner. Next slide, please. I'm almost done. So you can go in and dive deep into each metabolite. It provides you with the mass spec information, the structure information, the information on annotations that are available, connection to pathways. So it's rapid access to structures and downstream annotations from the structures. Next slide, please. So we have, in addition to everything else, we have this whole series of tools. We have hundreds of tools. I'm giving you a statistical toolbox list that we have on the workbench. By simply clicking on it, you can use your own data. You can use the data on the workbench. You can do normalization, univariate analysis, clustering and correlation, multivariate analysis, classification, comparative analysis. One of the most used resources on the metabolomics, world is the tools on the Metabolomics Workbench. And so next slide, please. Last 2 slides. So this is a kind of summary of what I told you so far. Metabolomics Data Repository is not just a simple resource. We provide the metadata information to allow you or enable harmonization, to enable deep studies, to enable deep analysis. And the only thing that I did not tell you, and which I'm going to tell you in the next slide, we need, if you're dealing with human subjects data, next slide, please. If you're dealing with human subjects data, you need clinical metadata, and this is absolutely crucial. And I know despite the privacy issues, the HIPAA issues, the consensual issues with reference to IRB protocols, despite all of that, we developed a minimal metadata standard that is now going to be adopted by the Common Funds Initiative, which deals with a very simple set of 6 tasks, 6 types of information that will provide the ability to harmonize data and to create knowledge graphs. For example, I can ask the question, find me all the species that are present between male and female, or between age, over 65 and less than 55? So those types of questions can only be answered if I have clinical metadata, and this is a basic scheme for clinical metadata. Next slide gives you a little bit more insight into how this clinical metadata is gathered and how it is organized. This is what we do in the Metabolomics Workbench. And so we essentially create a subset within this and make it computable. We are creating 5 profiles, which makes the metadata computable. And so this really tells you the importance of linking data and metadata and providing an infrastructure for navigating complex biomedical data. I don't have time to tell you today, but in addition to all of this, we also provide ability to translate this and integrate this with gene data, with protein data, and with all kinds of other types of data. So to me, as being a systems biologist, this is the holy grail of the future. We really need integrative approaches towards accessing data resources. With that, I will stop. Thank you for your attention. And a number of people in my group have been responsible for this. It's a very large number, so I don't have them listed here. And if you go to the Metabolomics Workbench website, you will find the list of all the people who contributed to the development of the Metabolomics initiative. Thank you again for your attention.
Great, thank you so much. Okay, let me get my windows oriented here. I had the sharing issue, so I was scared to click on anything while we were presenting. Okay, so I've seen a lot of chats pop up in the window. And so I think we are good to get started. So I want to ask if anybody within our attendees has a burning question that they'd really like to jump in with, or we can go through the questions as they were posted. Like I said, Marissa, I know you were tracking questions. Did you by chance get them?
So there's 2 questions that Lynn answered Lynn answered the first one and Scott answered the second one. Lynn said that the Mix S6 release will be posted today on Twitter, which is really exciting. Congratulations for getting that out. And then the next question Scott answered about how many of the defined reference materials have been used. And Scott, I don't know if you want to speak more to that, but I know you said that you're not really sure, but maybe less than 100. So I think that maybe we can move forward with the charge questions. And if anyone has any more questions, feel free to drop them in the chat.
Great, thank you so much. Yeah, so Scott, do you want to touch on that a little bit more?
Yeah, sure. So, I will say when NIST produces a reference material, we don't have manufacturing facilities like large biotech companies, New England Biolabs and ATCC, that produce these types of products all the time. We don't have those manufacturing capabilities. So we usually produce things on typically a smaller scale. And an example of that is this RM8376 that I discussed. When we manufactured it, we produced about 600 units. That's what we have to sell to you all. I suspect we've probably sold about 100 units so far. So, we've got maybe 500 left to sell. We'll sell them until they're no longer available. And then we will consider whether or not to make it again or make the next version of it, or, whatever next iteration that is. So get it while it's available, use it, and let us know how it works for you. Feedback is key here. We'd love to hear about how you're using it and how it's working for you, what works well, what doesn't work so well.
Thanks. All right, so kind of to get us started on these charge questions, we wanted to ask, in what ways do you use standards in your day-to-day work with the data types that you each work with? And we can start off with whoever feels really excited about answering this one. Okay, go for it, Lynn.
So, hi, Lynn Schrummel. So data standards are used throughout my daily life, I have to say. So I work at an institute that does all sorts of microbial genomics. We run the Human Microbiome Data Collaborative Center. I also work on the Metasub Consortium, where we're sampling subways around the world. So collecting data, any type of sample, and coordinating that with a submission to the NIST for the sequence data. We also just conducted a study on our annual meeting of soil and water in Morea. So we collected the standardized metadata of all the sampling sites so we compare the samples across a watershed.
So I'm happy to join and answer. So when you talk about standards, you need to parse standards into 3 or 4 categories of standards. Number one is the experimental technology and measurement standards, which includes all the mass spectrometry standards and so forth with measurements. And there is already substantial amount of knowledge and data available for these standards. And I would strongly urge folks to adopt these standards, which have been developed by a number of people who are experts in the field. Second set of standards deals with specific data associated standards, which includes following specific formats. And then in the mass spectrometry world, in metabolomics and proteomics, there are now very good emergent standards that people are using. And I would again urge people to use these standards so that it makes it interoperable and easily accessible. Third, there are standards with reference to nomenclaturing and naming of all the list of species, whether it's proteins or whether it's metabolites or whether it is something else. And so, or microbiome, names of microbiota. So to do this, in fact, some exist and some don't exist. As I mentioned to you, we developed one of the world standards, global standards, using RefMet, which really now serves as a universal standard for nomenclaturing metabolites. So there needs to be development in this area. Lastly, and but most importantly, there needs to be standards with reference to use of human subjects data and the metadata. And again, I refer to existing standards that people are developing and using, and not develop an arbitrary standard, arbitrary set of names, which makes it very difficult to interoperate between different types of data. And so this is where I want to bring standards as being extremely important in terms of adoption and development, the combination of both as we go along in this data work.
Thank you so much, Shankar. Scott, Mac, anything to add to those?
I'll just quickly add, a lot of the standards that we develop at NIST and work on at NIST are wet lab standards. So DNA standards, mock communities, human fecal material. We are working to produce these wet lab standards, and I will also point out that it's a resource for the users. ATCC has a suite of microbiome standards available, as does Zymo. So shop around on those websites, find the standards you need, implement them in your studies to show that your measurements are reproducible from day to day and your instrument is not drifting. I'll give you a quick example of where wet lab standards should have been utilized. There was I won't mention anything specific, but a 15-year prospective epidemiological study that was presented at a meeting a couple of years ago. They were collecting microbiome samples from infants over 15 years. At the time of this presentation, they were 7 years into the study, and they decided 7 years in that they were going to switch their DNA extraction kit and move to a different DNA sequencing technology. And in doing so, all of their data shifted accordingly because you've just changed your methodological variability. They did not have any in-house standards or bench, wet lab standards that they had been utilizing. So there was no way for them to really understand the impact that those methodological variable shifts had on their resulting data. So implement these wet lab standards early and routinely, and it will behoove you in the end.
And I'll just add, I agree with everyone's comments, but also one of the challenges we have is really educating people to use these standards and to implement them and to become aware of them, and especially in interdisciplinary communities where people are, maybe submitting to different kinds of domain journals. How do we really get the message out is something that I think is really important to think about.
Thanks. And that really touches on kind of just a closing thoughts as we're running low on time. So how would you encourage our audiences to engage with these standards, right? Like what's your advice to them if they want to expand this into their lab?
I think a good starting place would to look at literature and see what standards are out there for them. Both the EBI and NCBI have nice documentation on the standards that are used in different communities, and then see how they're utilized and see how you can then integrate them. again, as it was said earlier, reuse instead of reinvent if it's at all possible. Because I would say for most standards you need out there, there's someone who started on it. Find a community that wants to work on it with you if it doesn't already exist.
I had mentioned our consortium IMSA earlier, the International Microbiome and Multi-Omics Standards Alliance. If you go to imsa.org, we've set up that website to be what I call a one-stop shop for all things related to microbial standards. It is not just what IMSA is working on, but it's everything that we can find that's related to microbial standards, whether it's metadata or physical standards, etc. So visit imsa.org and hopefully you can find what you're looking for, links to other sites on that website.
Great, thank you. All right, we are at the top of the hour within like the next 20 seconds. So with that, I want to thank all of our panelists, Shankar, Lynn, Mark, and Scott for joining us. Thank you so much. And thank you to our attendees for being with us for this hour and a half. And we hope to see increased use of metadata standards in your research. And please don't hesitate to reach out to anyone within the NMDC if you need any help or guidance or have any thoughts. We are always looking for ways to expand and improve.
Thank you very much.
Thank you
so much.
Thanks.
Great, thanks everyone.